Hello everyone, welcome back. Today we are going to learn structure and functions of RNA along with their different types. First, you should know what is RNA. If you expand RNA, it is nothing but ribonucleic acids. So when you say ribonucleic acids, it is a nucleotides or polymers of nucleotides where sugar is ribose. Whereas in DNA, the sugar is deoxyribose. That is the second prime carbon atom, the OX or hydroxyl group is intact in RNA. And unlike DNA, the RNA is single-stranded. We know that in DNA contains two strands, whereas RNA is single strand. So when there is a single strand, there is no question of base pairing. Having said that, we will find base pairing in some types of RNA. When we deal with structure of different types of RNA, we will see which RNA has got base pairing. The bases present in RNA are adenine, guanine, cytosine and uracil. So instead of thymine is replaced by uracil in RNA. But there is an exception. This uracil can modify in certain types of RNA. It can be converted to thymine. When you go and watch the first video of this molecular biology, you can see the structure of uracil and thymine. Only the difference is methyl group. So when uracil is modified or if methyl group is added, it can be converted to thymine. It is called ribothymidiene because the sugar is ribose. In certain type of RNA, especially tRNA, we can see thymine also. But in general, we will see uracil in place of thymine. So this is a modified base. And same uracil can be modified to D. This is dihydrouridine. I will explain this structure when I explain tRNA. Another modification of uracil, it is pseudouridine. We represent like this. It is pseudouridine. So the major bases present in RNA are adenine, guanine, cytosine and uracil. Exception in uracil can be modified to thymine or D, that is dihydrouridine or pseudouridine. We will see what are the different types of RNA. There are three main types of RNA. First is messenger RNA. So the abbreviation is mRNA. The second type of RNA is transfer RNA. In short, we can write tRNA. The third type of RNA is ribosomal RNA. It is called RRNA. So these three RNA are the major types of RNA. Apart from this, there are many other RNAs. They are called minor RNA, that is heterogeneous RNA. It is also abbreviated as small h, small n, hn RNA. Another type of minor RNA is small RNA. Again, small RNAs are two types. One is small cytoplasmic RNA and another one is small nuclear RNA. So these are small RNA. One is present in the cytoplasm, another is present in the nucleus. Another class of RNA, they are called non-coding regulatory RNA. Again, there are two types. One is microRNA. It is abbreviated as small m, small I RNA. And another type of non-coding regulatory RNA is silencing RNA. It is abbreviated as small s, small I RNA or siRNA. So these from heterogeneous to non-coding, they are minor RNAs. These are the major RNAs. That means you should know the structure and functions of messenger RNA, transfer RNA, ribosomal RNA in detail. So we will see one by one. Now we will see the structure and function of messenger RNA or mRNA. So messenger RNA looks like this. So messenger RNA, as I said, it is single stranded and the base thymine is replaced by uracil. We will see uracil in mRNA. So the structure looks like this. Imagine this is a messenger RNA and it is ribonucleic acid, that means polymers of ribonucleotides, and all these are bases. Maybe here you can see A, here you can see U, here you can G, like that, U, C, G, like that. Okay. Hallmark or characteristic features of messenger RNA. In the 5' end, this is the 5' end, it is capped. The messenger RNA in the 5' end, it has got a base which is methylated and this is called cap, and the cap is called 7-methyl GTP, actually 7-methyl guanosine triphosphate. So the important or hallmark of messenger RNA in the 5' end, it has got G, which is methylated. So in the 5' end, this cap is mainly by is 7-methyl guanosine triphosphate. 7-methyl GTP is nothing but there is a G here which is methylated, and in the 3' end we will see lot of A. So this is called poly A tail. Okay, usually we will see 20 to 250 ribonucleotides here. That is why I have written in N times here. So this will become our 3' end now. Always if you take DNA or RNA, it runs in the direction of 5' to 3'. But the nucleotides, the phosphodiester bond is 3' to 5' phosphodiester bond. So the function of 7-methyl GTP cap or poly A tail is to stabilize the RNA or prevents exonuclease activity, or it helps in recognition of tRNA for protein synthesis or during the protein synthesis. So there are many bases present here and we will study in detail in transcription. Before transcription, these bases are grouped into 3 letters. They are called codons. They are called genetic codes. At this moment I will not go in detail. The mRNA or messenger RNA is a polymer of ribonucleotides. In the 5' end, it has got a cap, nothing but it is a guanosine nucleotide, ribonucleotide, and which is methylated, 7-methyl GTP. And in the 3' end, it has got large amount of A adenosine residues, polymers of adenosine residues. They are about 20 to 250 adenylate we can see in the 3' end. It is called poly A tail. The functions of mRNA, its function, it serves as template for protein synthesis. That means this mRNA, the formation of mRNA is called transcription. Whatever codes are present in our DNA, it is rewritten. The double-stranded DNA is rewritten as single-stranded DNA. So these codes are actually complementary to the codes present in the DNA or particular gene. Now this mRNA serves as a template for protein synthesis. That means the ribosomes which is present in the cytoplasm will utilize these codes or genetic codes written in the mRNA. With the help of tRNA, there will be synthesis of protein. That process is called translation. We are going to study all these processes in coming videos. At the beginning, you should know messenger RNA is a messenger to transfer the genetic information from the DNA. So this is the brief explanation about the structure of mRNA. Now we will see the transfer RNA. Transfer RNA or tRNA, there are 20 different tRNA. We learned in amino acid classification, we have 20 standard amino acids. That means we have 20 transfer RNA for 20 amino acids. tRNA might contain about 74 to 95 ribonucleotides. Whereas messenger RNA structure is varies because it mainly transfer the information. It depends upon the gene. Whereas transfer RNA usually it contains 74 to 95 ribonucleotides. And you should remember that 10 to 20% of this tRNA contains modified bases. So in the beginning introduction I told uracil can be modified. So 10 to 20% of bases present in the tRNA are modified. So this modification is mainly from the uracil base. This is a uracil actually. Now the difference between uracil and thymine is very minimal. If methyl group is added here, it will become thymine. And here if instead of deoxyribose, if ribose is added here, so it will become ribothymidine. So it is an unusual base present in the tRNA. So the name here it is called ribothymidine. Usually in RNA we will not see thymine, instead it is replaced by uracil, but sometimes especially in tRNA there is thymine, that is modification of uracil. There is addition of methyl group, so it will become uracil will become thymine. It is this base is present, unusual base is present in tRNA. So another unusual base is dihydrouridine. Again this is a modification of uracil. This is a uracil here. Here the double bond is reduced because there is addition of hydrogen here. So this is called dihydrouridine. Just modification of uracil base. And the one letter abbreviation for this base is D. Another modified base from uracil is pseudouridine. It looks like uracil but it is somewhat different. Usually nitrogen atom in the first carbon atom. This is the first carbon atom. But here in this case pseudouridine, there will not be nitrogen atom. This is carbon actually. And then it will be attached to ribose. The first atom of pyrimidine is nitrogen in uracil or in all pyrimidine base. Here it is carbon. So we will see carbon-carbon bond especially in pseudouridine. Usually we will write U and put like this. This will become pseudouridine. It has got oxygen in second carbon and fourth carbon as that of uracil, but in the fifth carbon atom instead of carbon it has got nitrogen, and in the first carbon atom instead of nitrogen it has got carbon. These are the modified bases we could see in transferred RNA, that is D, pseudouridine and T. We will see the structure of tRNA now. The structure of tRNA or transfer RNA, it looks like clover leaf appearance, and the structure it has made up of major 4 arms, and also in addition to this main 4 arms it has got one variable or extra arm also. So let me draw the structure of tRNA. It has got mainly 4 arms. The transfer RNA looks like this. So this will be the 5' end and this will be the 3' end. And as I mentioned, it has got 4 main arms. At the 3' end, we have from 5' to 3' we have in the 3' end we have CCA. That means cytosine and adenosine nucleotide. And this arm is called acceptor arm. So why it is called acceptor arm? Because it actually accepts the particular or specific amino acid and it carries to the site of protein synthesis, that is ribosomes. So when mRNA which carries specific information in the form of genetic code to the ribosomes, this transfer RNA carries activated amino acid specific to that particular codes or codons present in the mRNA. So it accepts that particular amino acid for protein synthesis. That is why it is called acceptor RNA. It is present in the 3' end. And always the 3' end, the bases are CCA from 5' to 3' direction. Another arm, as I mentioned just 5 minutes ago, in tRNA there are modified bases. So Here in this we can see there is a loop. We will find the modified base D, that is dihydrouridine. So this arm is called D arm and the loop is called D loop. Okay, this whole arm is called D arm and the loop is called D loop. Similarly, the bottom side will see a loop or arm. It has got bases and these bases are complementary to bases or codes present in the mRNA. So the bases present in the mRNA we grouped into 3-letter bases. They are called codons. Whatever bases present in the mRNA, exactly complementary bases are present in this tRNA, especially in this arm. So this arm is called anticodon arm. Okay, remember why it is called anticodon arm? Because these bases which are present in the anticodon arm are complementary to the codes present in the mRNA. Now, again I explained unusual bases. Here in this particular loop we will see T. Usually we will not see thymine in the RNA, but in tRNA we will see thymine just modified from uracil. Methyl group is added, it will become ribothymidine. And also we will see pseudouridine, that is again modified uracil, and cytosine. So this arm is called T pseudouridine and C arm because of presence of modified bases. So these are the 4 major arms present in the transfer RNA. In addition to this, we can see this arm. This is called extra arm. It is not a constant. It is different for different tRNAs. So that is why it is also called variable arm. In the beginning of this presentation, I told all RNAs are single-stranded. There is no question of base pairing, but there is exception. So this single-stranded RNA, especially tRNA, they form a loop here, hairpin loop. So they will form base pairs with the complementary bases here. So usually in the acceptor arm we will see 7 base pairs, whereas in anticodon arm we will see 5 base pairs, and also this thymine and pseudouridine arm also we will see 5 base pairs, whereas in the D arm we might see 3 or 4 base pairs. Remember, this is not a double-stranded RNA, it is a single-stranded RNA, but it is placed in such a way that we can call it as a secondary structure. They will form a loop and complementary base makes the base pairing. So this is the structure of tRNA. What is the function? It carries amino acid to the site of protein synthesis. So this is the functions, carries amino acid to the site of protein synthesis. Site of protein synthesis is ribosomes. So now we studied the structure of tRNA. Again, the detailed function we will study under protein synthesis or translation. The next important major RNA is ribosomal RNA or rRNA. So this rRNA, the name suggests that it is present in the ribosomes. We all know that ribosomes are particles, a subcellular, it is present in the cytoplasm. When it is present in the endoplasmic reticulum, we call it as rough endoplasmic reticulum. Ribosome contain actually made up of RNA and protein. So ribosomes are nothing but it is combination of RNA and protein. So we can call it as nucleoprotein. Nucleoprotein doesn't mean that it is present in the nucleus. Nucleo means nucleic acid. Proteins and nucleic acid. It is called nucleoprotein. Even our DNA is also nucleoprotein because it contains deoxyribonucleotides and proteins, histone proteins. Whereas ribosomes are present in the cytoplasm. It is nothing but combination of proteins and RNA. So these ribosomes are mainly a factory site or a machinery for protein synthesis. Protein synthesis takes place in ribosome. The major function of ribosome, it helps in translation of protein synthesis. So the ribosomes are made up of 2 subunits. It has got 2 subunits. So one subunit is called large subunit Another one is called small subunit. And the subunits usually we designate as a unit called Svedberg unit. It is designated as S. That means if you take these ribosomes and subject under ultracentrifugation, so they will measure their sedimentation coefficient. Okay, so this sedimentation coefficient is designated or measured in terms of Svedberg units. We call it as S. I will write this structure of ribosomes for both prokaryotes and eukaryotes. So here I have written ribosomal RNA for, this is for prokaryotes and this is for eukaryotes including human beings. So I already mentioned the units represented as S or sedimentation coefficient. In prokaryotes we call the prokaryotic ribosomal RNA, it is 70S, whereas in eukaryotes it is 80S. So prokaryotic, the large subunit, so I'll write here, this is the large subunit and this is the small subunit. So in prokaryotes, the large subunit is 50S, this is 50S, and the small subunit is 30S. And you should remember this 50S and 30S is not, you should not add. The together sedimentation coefficient of prokaryotic RRNA is 70S. So you should not add 50 plus 30. It is a sedimentation coefficient or Svedberg unit. Similarly, in human beings or in eukaryotes, the large subunit is 60S and the small subunit is 40S. Here also you can see if you add 60 plus 40 it will become 100, but together it has got only 80S. As I mentioned, the ribosomes are nothing but RNA and protein. So ribosomal RNA is most abundant RNA because you can see in prokaryotes there are mainly two types of ribosomal RNA. Here we in prokaryotic large subunit we have 23S RRNA and we have 5S RRNA. That means there are two types of RRNA present in the large subunit of prokaryotic ribosomes. And we already mentioned they are nothing but combination of RNA and proteins. So here in the large subunit, in addition to this RNA, they also have proteins. So that's about 34 proteins. And the small subunit also has got RNA. It is 16S RRNA, ribosomal RNA. And here also we see proteins, around 21 proteins here. In eukaryotes, we have, in prokaryotes in the large subunit we have only two types of RRNA. But in eukaryotes in the large subunits we have three types of RRNA. First one is 28S RRNA and 5.8S RRNA and 5S RRNA. And in addition to that, eukaryotic large subunit has got about 40 proteins. The small subunit also has got 18S RRNA and the proteins about 30 proteins. So this is the structure of ribosomal RNA. So functions you already know that it is the site of protein synthesis, and in addition to this, ribosome or ribosomal RNA also acts as an enzyme. It is called ribozyme. So this ribosomal RNA, especially RRNA, the major function is for protein synthesis and it also has got enzymatic activity. The first function is protein synthesis. The second function is it functions as an enzyme, ribozyme. It has got catalytic function also. So this is with respect to rRNA or ribosomal RNA. These are the major RNA. Coming to others, minor RNA, we have what is called heterogeneous RNA. So this heterogeneous RNA is just a precursor of mRNA. If you take a cell, it has got nucleus and in the nucleus we have DNA. We know that RNA is synthesized from the DNA. It is called mRNA. The initial RNA or mRNA, which is produced from the DNA, that is process is called transcription, is actually called heterogeneous RNA. When heterogeneous RNA, it will be modified depending upon the gene, okay, and there will be addition of cap and there will be addition of poly A tail, then it will become mRNA. And remember, the heterogeneous RNA is very large RNA. It can be spliced into different types of mRNA depending upon the gene encoding. For example, if you take in intestinal mucosal cell, we have apoprotein B48, and in the liver we have apoprotein B100. So this is mainly by splicing, the conversion of heterogeneous RNA to different RNA. If you take different types of immunoglobulins, there also it is initially heterogeneous RNA, it will be converted to different type of mRNA and different type of proteins are synthesized. And it will be stabilized by adding cap at the 5' end and poly A tail, it will become mRNA. So this mRNA actually initially synthesized in the nucleus, it will come out to the cytoplasm. So another type of minor RNA is small RNA, that is small cytoplasmic RNA, which is about 294 ribonucleotides, and it mainly helps in recognition of signal sequence. That means it helps in protein synthesis, recognition of signal sequence in protein synthesis. I am not going to explain here, we will see in detail when we study translation. Another type of small RNA is small nuclear RNA. It is also about less than 300 ribonucleotides, and it's mainly helps in processing of RNA. It's very helpful for a process called splicing, that means removal of introns, that we will discuss in transcription. And it also made up of different components, they are called U1, U2, U4, U5 and U6. Okay, we will study in detail when we study transcription. Another type of minor RNA, so it is called non-coding regulatory RNA. Here there are two types. One is microRNA, the abbreviation is mRNA, and another one is silencing RNA, the abbreviation is SI. So both these are non-coding RNA. Actually they helps in gene expression, in fact they inhibits gene expression. So I am not going in detail for other minor DNA. You should know the major RNA, that is messenger RNA, transfer RNA and ribosomal RNA in detail. So this is with respect to structure and functions of RNAs. If you want any simple notes with respect to the structure and functions of RNA, please visit my website. The link for my website I will put it in the description box below. If you like this video, please give a thumbs up. If you haven't subscribed so far, please subscribe and press the bell icon for future notification. Thanks for watching.